Hello student, welcome back in one more video. Friends, as we know that this is the sequence of the chapter sexual reproduction in flowering plants. In last video, we will briefly discuss about the parts of flower and the two major roles that is present there. One is essential and another one is non-essential. Already we had discussed why one is essential and another one is non-essential. Now in this video, we will going through the topic in detail. Pre-fertilization event. So what is pre-fertilization event? As we know, fertilization means fusion of two opposite gametes. Gametes are the haploid reproductive cell. As we know that the angiospermic plant is diploid. Suppose if two diploid cells are fused, then the tetraploid progeny is produced. The complete genetic makeup of the organism is completely altered and the progeny that produces is not same as the parents. So, before fertilization, before fusion, there is a very important event that should occur, that is pre-fertilization event. What is the name of that pre-fertilization event that occurred before fertilization? Exactly, the event is gametogenesis. Gametogenesis means production of haploid gamete from the diploid cell. How this gamete is produced, we will going through the heading microsporogenesis. By this process, the male gamete, the haploid male gamete, that is microspores, is produced, and the second is megasporogenesis. By this gametogenesis process, the haploid embryo sac is produced. Now this two topic is very important as for the aspect of class 12 as well as for NEET examination. Many question occur from this portion every year. So let get explore this topic. First we will going through with microsporogenesis. So let's get start. Guys, so to understand microsporogenesis in detail, first of all we have to remind the structure of male reproductive part. So as I told you, the male reproductive part of flower is called androecium. But androecium is the name of the group of male reproductive part. What is the name of single unit? Exactly, the name of the single unit is stamen. So the group is androecium. The group is androecium, but the unit is stamens, right? Suppose if I am talking about that there are 5 stamens in a flower, so collectively it called androecium, single androecium. But how many stamens are there? So there are 5 stamens. Hope you understand that this is the collective part is called androecium, but the unit is called stamen. Now let's understand the structure of androecium to reach the process gametogenesis. That is very important. So let's remind the structure of stamen. As we know, the stamen is made up of two parts. The first is anther lobe, and the second is elongated portion that hold the lobe, that is called filament. The basal elongated portion, that is filament, which digital and hold the two anther lobe. Now the anther lobe and the elongated portion is filament. The structure of this stamen. Now we make the structure of the stamen like this. There is the elongated filament that hold two lobes together like this. So, and there is a connective. What is this connective means? The connective means that the vascular connectivity that is going to anther lobe, the vascular connectivity through which the plant takes the food and minerals, water, everything, that is that connective, the vascular strand. That connective, it means it is the vascular strand. Now this filament, as I told you, it holds two anther lobes. Two lobe, that is anther lobe. Okay, now the most important thing that to explain anther lobe, this is bilobed. What is meaning of bilobed? As you see that there are two lobes of anther, so we define the anther as bilobed. Bilobed. lobe. The second term to define anther, that is tetrasporangiate. Tetrasporangiate. Tetra, the meaning of tetra means 4 and sporangiate means if there is 4 sporangia inside the anther lobe, that is called tetrasporangiate. That we will see later in the diagram. But the most important thing that you have to understand with this portion is the diagram. That diagram is most important to understand. Then you can going through any of the question from this section, and this is very important. Probably the question will come from this section every year. So you have to understand it clearly. First of all, the one term is also used, that is dithecus. What does it mean, dithecus? Dithecus is also very important term to explain the anther. Means what is dithecus? Now try to understand what is the dithecus. And one more thing, that within the theca there is a very deep groove that is going through each lobe. This groove I am talking about. This groove, I am very sorry for that, but I have no color pen. This groove, there is a one longitudinal groove that is going through with both of the lobe. This groove. Now try to understand what is dithecus, how it is tetrasporangiate, and I hope that you will understand why it is bilobe, because there is 2 lobe is present. That's why anther is bilobe. Now try to understand why it is tetrasporangiate and dithecus. Then this diagram is most important to understand the term bilobed, dithecus, tetrasporangiate and connective. Everything you will understand very clearly after understand this diagram. So the first diagram is the diagram of stamen, and hope you all understand that this basal elongated portion is filament and the upper lobe like structure is anther lobe. Is it clear to you? And that longitudinal groove, this is the longitudinal groove. Now suppose if we cut this stamen like this, the TS, the transverse section, if we cut, normally we cut the anther lobe of the stamen like this, so we will found a clear cut 3D structure like this. This is the 3D structure. Now understand the difference between the stamen, this diagram and this diagram. You try to understand with this ink bottle. Now this is the 2-dimensional figure and this is the 3-dimensional figure. What is the basic difference between this figure? Try to understand with this ink bottle. This is the 2D structure that you can see. Suppose if I cut here as a transverse section, and if I show you like this, so we will get the complete structure of this. Suppose if this is the stamen and if I cut from here and look the 3D structure, like simply this is the structure of the stamen. If we cut here, this is the 2D structure, but if I will show the 3 dimensions, if you are able to see this 3 dimensions, so this is the basic difference between this figure and this figure. This is also the figure of a stamen and this is also figure of a stamen, but this is the 2-dimensional structure of a stamen, and after transverse section cutting, this is the 3D structure. So what you will find inside this stamen. Now friend, try to understand, this is the connective, here this is the connective, and this is the groove that is going through longitudinally. Now this groove will only separate the theca. Now where is the theca? Can you able to see the theca in this diagram? No, we will find the theca in this 3D diagram. That's why my motive to explain, that's why I make this diagram. Now this is that longitudinal section, this is sterile, but within this there is one theca and this is second theca. This is theca number 1 and this is theca number 2. Now try to understand, compare this figure with this figure. Suppose this is your first lobe and this is your second lobe. This is your lobe number 1 and this is your lobe number 2. Now compare with the 3D diagram. This is your lobe number 1 and this is your lobe number 2. Hope you understand this diagram. Now this is that sterile elongated tissue, that groove. This is the groove. Now this group separate two theca, and it is very clearly written in NCERT that there are two theca in one lobe. Two theca in one lobe. So this is the lobe number 1 and this is theca number 1. This is theca number 2. This is theca number 1 and this is theca number 2. So as same in lobe number 2, this is theca number 1 and this is theca number 2. Now the third term that we have to understand is tetrasporangiate. What is the meaning of tetrasporangiate? Tetrasporangiate means 4 sporangium is present in a typical angiospermic anther lobe. Now, sporangium is the place where the spores are produced. So the spores are produced inside this microsporangium. So how many microsporangia are there? 1, 2, 3 and 4. That's why this structure is tetramicrosporangiate. Hope you will understand these 3 topics. One is bilobe. Why it is bilobe? Because 2 lobe is already present in this stamen. Why this is dithecous? Dithecous because there are 2 theca that is present in each lobe. And why it is tetrasporangiate? Because there are 4 sporangium that is separated through that longitudinal group, 4 sporangium, which can produce the spores. That's why it is tetrasporangiate. Hope it is clear to you. This third diagram is the structure of microsporangium. We will discuss this in next video. Thank you so much for watching.